between knowledge and understanding. Knowledge is often synonymous with information. It is important, but I do not put a very high premium on you gathering a lot of information. It is good to have, but it is not the determinant for excellence. I put a premium on your ability to understand. Okay, so therefore, this 2 hours will be the typical exam length when you will be able to, think about the questions. Okay, so you can work it out on your exam spreadsheet or like on your workbook or whatever. And then when you come to the Viva, you will have to explain it to me, and that time the video will have to be on. Okay, so that's how it will be done. So now I'm coming back to Shayan's question. So Shayan, the function is actually defined by the design of the genetic screen. So all of you should understand that for a genetic screen, the design is everything. The rest is mechanical. It is the same thing for no matter which purpose you are conducting a genetic screen. It is always the design. So in the given example, what was the design? That yeast that cannot grow in presence of galactose as the only source of carbon. So obviously the mutants that won't be able to grow in presence of galactose as the only source of carbon, they will be defective in that pathway only. Of course, there will be few exceptions, but generally speaking. Therefore, after you do complementation analysis, mutants belonging to the same complementation group or different complementation groups, they should all be responsible for galactose metabolism one way or the other. But those who fail to complement each other, meaning belonging to the same complementation group, chances are that they belong to the same gene. They are mutants in the same gene. Is that clear, Shayan?
Yes, sir. I was just, can I ask again that, so here our target is that whether one gene is responsible or more than one gene is responsible for a single pathway, let's say for galactose. Yeah, that's our target.
No, that is not our target. That is the outcome of the analysis. You did not set out with the question that whether one gene is responsible for galactose metabolism or 30 genes are responsible for galactose metabolism. You only set out to discover, can I find out gene or genes responsible for galactose metabolism? You found 100 mutants, then you did complementation analysis, like extremely rare, unheard of, that all 100 belong to the same complementation group. You say, okay, there is only one gene involved in the pathway, but more likely you figure out 12 complementation groups. You predict that at least 12 genes are involved in the pathway. Is that clear?
Yes, sir. It's clear.
Okay, so now let me ask people who have kept complete silence so far, and I would request you to please do not stay silent. Okay, it is very easy for you to stay below the radar, but exams will be the same for all of you. Please interact. Okay, I am happy to give more time than needed, than officially allocated, but you must take advantage of it. Okay. So my question is to Adil Raja. Would you please turn on your video, Adil? Adil, are you there? Adil, are you there? Okay. Swagata Sarkar, would you please turn on your video?
Hello, sir.
Yeah.
Sorry, sir. I can't turn on my video because there is a internet problem due to heavy raining here. So
Fine.
my audio will be not be heard.
Fine. You tell me in complete detail the screen to identify mutants of galactose metabolism pathway. How would you do it? Let me break it up for you. What will be the starting yeast strain?
So,
Yeah, answer please.
sorry, sir. Can you ask the question one more time? I can't hear your voice. It's
To conduct the mutagenesis screen for galactose metabolism pathway, I want to know the full detail. Start with what will be the yeast strain that you will start with.
sorry, sir. I don't know about it.
Why don't you know about it? Did you listen to the lectures?
Yes, sir.
How many times? Should I play
Uh,
it for you where it is stated?
uh,
See, the advantage of this video class lectures is that everything is on record.
and sir, about the lectures in IITK. Hello, IITK.
Hmm?
Is that in the lecture in Hello IITK?
Either Hello IITK or the YouTube channel, these are the same lectures. It will be there in lecture 3 and lecture 4. Okay, anybody in the class would like to volunteer? Okay, Ayush, I will come to you. Just give me a minute. Ayush, bolo.
Sir, I have to answer the same question, right?
Yeah.
Okay, so what we will do is we will take a wild type yeast strain and we will mutagenize it using XYZ mutagen, whatever we are using for yeast, and then we will plate the mutagenized strain on.
So stop there.
Yeah.
Wild type is strain if you write in the exam or if you state that in the viva voce, I will not be able to give you more than 10% of the allocated marks.
Okay.
You have to define the wild type yeast strain.
Okay, which one?
Yeah,
So W303.
haploid or diploid?
Okay, that is the question. We will take a mm, we will take a haploid strain.
Why shouldn't you take diploid?
If we are mutagenizing a diploid strain, then the recessive mutations won't be screened. I mean, if the mutation is a recessive in nature, then we will not be able to characterize without tetride dissection that is it a haploid or a diploid mutation.
Okay.
That is why we have to take a
Okay.
haploid strain.
I will supply the language to you. Listen to it, and of course, I will share the recording. If you start with a diploid strain and the concentration of mutagens, the way they are used is to minimize the possibility of multiple mutations in the same cell. Because of these two, one yeast cell is likely to receive only one mutation. So in a diploid strain, therefore, the other copy of the gene in the sister chromosome will remain wild type. Therefore, any mutation that is recessive in nature will not display any phenotype. This is the reason why you should not use a diploid strain, but you should start with a haploid strain.
Yes, sir.
Right? Is there anyone in the class who do not understand this concept?
Understood.
Akanksha, go ahead.
When we began the experiment, like we began by saying that we choose this wild type yeast strain, and then we mutagenize it. So when we are mutagenizing it, like, is it a random mutagenesis?
Akanksha, for heaven's sake, do not say wild type yeast strain. Say wild type haploid or wild type diploid.
Okay.
Okay, make it part of your thought process. Okay, never deviate from that.
Okay.
Now go ahead.
Yeah, so a wild type haploid strain. So when we are taking it and mutagenizing it, so is this mutagenesis random, or is it like we know where to mutagenize it?
Random, completely random. So what you are doing is you are introducing these, you are feeding the yeast cells on these chemicals that typically cause formation of thymidine dimers. Okay, and that results during the replication, that results in misincorporation of a nucleotide.
Okay.
Okay. This, I'm sure you have studied about it earlier also, like UV related mutation, cancer, etc. This is the same mechanism everywhere. Okay.
Sir, should I ask a short question? Sir, in the video where this screening was told, the first they were mentioning MAT alpha and MAT A type of yeast. So what are these MAT A and MAT alpha type, and are they related to any this kind of screening? Like, I didn't understood the concept of MAT A and MAT alpha.
Okay. So I have shared a folder called resources. Have you seen that?
No, sir. Maybe I have missed that. I downloaded that yeast paper which you gave with the third lecture.
Just let me see what I have with the third lecture. No, that is the lecture slide, right?
No, sir, there is one paper, I guess. I can read the name. That is The Art and Design of Genetic Screening Yeast.
ठीक है. So you have that, right?
Yes, sir.
Can you also look at Hello IIT another time? Can you open that?
Yeah, sure, sir.
Please open it once.
Yeah, sir. I have opened it.
Do you see any resources folder there?
No, sir. There is one resource option, but inside that I don't see anything.
Okay, inside that you don't see anything. Okay. Okay. So where you see art design of Yeast Genetics screen, do you see another one, Yeast Genetics Fred Winston 2002?
Let me check, sir.
Sir, I downloaded that particular paper from the link given beside lecture 3.
So that's the thing. You need to read that, okay?
Okay, sir, I missed it.
Just as background, Fred Winston is one of the most celebrated yeast geneticists in the world today. Okay? And I happen to be in the same department as him. Of course, he was a professor and I was a postdoctoral fellow. So on my request, he shared his lecture material with me, which I have shared with you. So you read that very carefully. It is, of course, like you should, like this is what I tell every batch of students, that if you want, our Prime Minister talks, it cannot happen unless you have Atma Samman. You must have pride in yourself. That is the course material that PhD students at Harvard studies. Give me one reason why I should not hold you at the same level. Yeah, I want to hold you at the same level. So you need to work hard. You need to study. I will facilitate. If you study, I will facilitate. Now, coming to your question. MAT is a small form of mating type. M-A-T-I-N-G, mating type. Just like humans, where we have male and female, you can think of it as mating types because male mates with females. Similarly, in yeast, there are these two mating types, MAT A and MAT alpha. Only during mating of two haploids, MAT A cannot mate with MAT alpha. MAT A cannot mate with MAT A. MAT A can only mate with MAT alpha. And I will teach you in the context of transcriptional regulation how the mating types actually can change. Unlike us, in yeast, the mating type can change. It is called mating type switch. Okay? But the reason these mating types are mentioned is to allow you to understand that haploid you are going to make diploid, you have to use trains from opposing mating types. Okay, you can like say you have done your screen and you have 100 mutants, you want to do complementation analysis. You started with W303 alpha, which is mating type alpha. So you cannot do any complementation analysis. So you will have to change the mating type for every pair that you would like to test for complementation analysis. And that is a rather straightforward process. We will come to that later. Okay. Does that answer, Piyusha?
Yes, sir.
Okay. Anything else?
Sir,
No.
I have a question. Like,
Mhm.
sir, like in once while we are introducing mutations in yeast, like the formation of pyrimidine dimers which are taking place in E.
Mhm.
coli, there are like repair mechanisms such as photoreactivation. But in
Mhm.
yeast, like, does it work that way, or do they have a DNA repair mechanism, or is it prominent, more prominent in the wild type strains which are available?
No, yeast of course has a DNA repair mechanism. Hmm. But so that see, like when you talk about frequency of mutation, you talk
Yes, sir.
about it after taking into account the repair mechanism, because repair mechanism is of course there. You also have repair mechanism. Every human being also has repair mechanism. Even after that, cancers do happen, right? So that's
Yes,
when
sir.
the repair mechanism is overwhelmed or it misses it.
Okay, sir. So, like, is the duration more prolonged in yeast? Like, if we are introducing, so how long does it take to revert back the mutation which actually takes place?
No, no, no. Once it is mutated, it is very difficult for it to revert, because for all you know that
Okay,
that's
sir.
a new sequence that is thinks its own, right?
Sir, a follow-up question
Yes, sir.
from Samaj's question. So the thing is, like, if we are using the mutagen and introducing the mutagenesis, now, like, out of the 100 in all the cells, the mutagen has gone, but it may be like because of the repair mechanism, it is coming, back to the again in the yeah.
Yeah.
So only like 30% or 40% will be affected.
Mhm.
Is that the situation or something else happens?
Okay, if you really want to know, I will tell you how exactly it is done.
Yes, sir.
Yes,
So
sir.
you need to titrate the amount of mutagen, the quantity of concentration of mutagen. Okay,
Okay.
uh, for hydroxyurea or whatever you are using. How would you do it? It's a purely mathematical calculation. I will make it easy for you to understand by giving a commonplace example. Let us say this I am giving you only for example's sake. This is not how the classical yeast genetics was done. Okay,
Okay, sir.
but you will get the concept. Let us say that you made an yeast strain W303 alpha, but it also expresses GFP.
Okay.
Okay, so it will be a green colored yeast cell, right?
Yes.
Yes, sir.
Now GFP has 750 bases.
Hmm.
So you know for the overall length of the yeast genome, 750 is which fraction of it, right?
Yes.
Yes, sir.
Now you treat it with the mutagen and you see that in what fraction the GFP is not there.
Okay.
Okay, if it matches more or less with the fraction in terms of size, like 750 by 10 million, huh? Then you know that the GFP at that concentration, the GFP ORF is being mutated at least once.
Okay, sir. Not very much clear, sir.
Okay, okay. So let us try to do it this way. You think of a football field, okay? A very large football field, and you only have the ball at the center of it, okay? Now you are allowed, just because I am allowing you to, you can throw projectiles inside the field. You have to hit the ball.
Yes.
Assuming that you have zero ability to target, what will be the frequency with which your projectile will hit the ball? It will be the ratio of the surface area of the football versus the surface area of the field, right?
Yes, sir.
Yes, sir.
Now, instead of the ball, you think of the central circle.
Yeah.
Now the ratio will increase, right? That
Yes.
is, your projectile will hit the central circle
More
more often.
frequent. More often.
So, and this ratio will always be dependent on the size ratio of the field and the central circle, right?
Yes.
Or the area that you are defining, right? Now think of the yeast genome as the football field
Okay.
and GFP as your defined area.
Yeah, ball at the center of the area. Yes.
So you know the ratio of the nucleotide length between whole genome and the GFP, right?
Yes.
So
Yes,
if
sir.
it is completely random, then you will get the number of in a large population, the number of yeast cells that will be hit in the GFP will be exactly in the same ratio as the nucleotide length ratio.
Yes.
So when you find that in a large population, the cells that have become GFP negative in the same ratio, then you know that your mutagen concentration is such that mutation is happening only once in the cell.
Okay.
Did you get
Okay,
it?
sir.
Yes, sir.
Yes, sir.
Now there is a caveat in it. The caveat is that there can be someone's sound is on. There is a background noise coming. Ankit, maybe your sound is on. So the caveat is that there will be certain base changes that will be silent mutations. So GFP will still be on. There will be certain base changes which changes an amino acid, but it still does not affect GFP protein structure and therefore fluorescence. So it will still be an overestimation, but it is an overestimation that you can live with. Okay?
Okay, sir.
Everybody understood?
Yes, sir.
Yes, sir.
Okay. So now you have done the mutation on a haploid strain. Now, Adil Raja, are you back? Okay. I got the first case of sleeping student. It happens online as well. Sandharv, are you there?
Yes, sir.
After you have done the mutation, so please close your eyes and think. The mutagenesis, have you done it in a liquid suspension of the yeast cells or on a plate?
Plate.
No, sir. If you are doing it on a plate, at max, you will be able to mutate 300, 400, 500, 1,000 yeast cells at a time. Because yeast cells, just like bacteria, grow in colonies. Okay. So in a plate, you can have, so many colonies. But if you are growing it on a liquid culture, every yeast cell is an independent entity. Therefore, you will be able to hit millions of cells simultaneously. Okay? Anyway, so you will mutagenize in the liquid culture. After that, what will you do, Sandharv?
Hello.
So you will mutagenize in liquid culture. After that, what will you do?
Sir, I thought you were saying you were. I mean, you were not asking. Can you please repeat?
Yeah, I said that you will do the mutagenesis in liquid culture. Now the question is that after you have mutagenized while the cells are in liquid culture, what would you do next?
So I'm not understanding what like in what context.
Okay, it is in the context of yeast mutagenesis screen, sir. Anupam Mishra,
Yes, sir.
what will you do next?
Sir, we will try to identify the mutations.
How?
Uh, so,
The cells are in a liquid suspension, right?
yes, sir.
How would you identify the mutants?
So we need to separate the cells.
How? Okay, my indication is you guys are not paying attention.
Sir, can I try?
Yes, Shubhajit, please
Yes,
go
sir.
ahead.
Yes, sir. First of all, we have to take haploid yeast cells and which can grow in presence of galactose. So we can call them as wild type. And as you have already mentioned that we have titrated, titrated in such a way that after adding mutagen, the mutagen will cause mutation, only one mutation in one cell, right?
Yes.
And then after incubating with the mutation, then we have to plate the haploid yeast in the media containing glucose.
Very good.
Then after the incubation, we can, as we are going to the analysis for the galactose mutation pathway, then we have to do a replica plating of the plate which we have already grown in presence of glucose. And then we have to, after replica plating, in the replica plate we will not add glucose. In the replica plate we will going to add galactose. And then we compare what are the number of colonies, what are the changes in the number of colonies between the plate which are going in the glucose and which are going in the in presence of galactose. So those cells which are absent in the galactose but in presence, those colonies which are present in glucose but absent in galactose, we can pick those colonies from the plate of glucose, and we can go further analysis for the galactose mutation. We can from this analysis we can confirm that any one of the gene involved in the galactose metabolism may be mutated in those colonies.
Excellent, Shubhajit. Excellent. I'm very impressed. Ayush, you were saying something.
So I have a doubt. I mean, if we are screening for the mutants for galactose metabolism, so why are we even using glucose here? We should simply grow them in the presence of glucose in one plate and then replica plate it in a media without the galactose as a carbon source. So the colonies which will not be growing on minus gal plates will be the ones which are mutated for galactose metabolism, right?
So how is that different from what Shubhajit said?
He said that he'll be plating them on a media containing glucose and then replica plating on a plate having galactose as a carbon source. So what we'll be getting is on the galactose media plate, all the colonies which are either mutated for galactose metabolism or not, because there will be galactose as a okay, I get it now. Sorry. So we will get the colonies which will not be able to utilize galactose as a carbon source. Yes, I get it now.
So Shweta Kushwaha.
Yes, sir.
Why would you need to plate in presence of glucose?
What plating on galactose plate instead of glucose?
So both Subhajit and Ayush say that from the liquid culture where you are growing the yeast cells in the presence of the mutagen, you will first plate them on glucose containing plate.
Yes, sir.
Then you will shift them by a replica to a galactose containing plate. Like, to be precise, a plate that will have galactose and not glucose.
Yes, sir.
My question is that since you are looking for mutants that are defective in growth in presence of galactose, why would you at all plate them in presence of glucose?
So, because when we are plating it in galactose medium, then that particular enzyme, which is now mutated, will not convert galactose into glucose, as glucose is used as energy source. If we are plating that colony in a plate containing glucose, then glucose is already present in the medium. So, it will not require any other enzymes to break down galactose to glucose, as glucose is already present here.
Okay, if you write this answer, or if you give me this answer during Viva Voce, I will have no option but to give you a sweet 0. Okay. I appreciate structured thinking. Please listen to the lectures as many times as you feel like and start to think. There is zero benefit out of remembering the lectures or remembering the book pages. Okay, start to think. You try again, Shweta. Your answer is absolutely wrong.
Okay.
Sir, I have some questions.
No, I'll come to you, Shubhajit, in a minute.
Okay, sir.
Nageshwar, bolo. Why should you?
I was like,
huh?
Yes, I was wondering if the plating in glucose would be necessary, because we can directly go with plating the yeast cells in galactose itself in the first place, because we are like screening the mutations that are only involved in the galactose metabolism pathway, right? So like in the first phase, like we can, after mutagenizing the yeast cells in liquid culture, we can first plate it in the plate containing galactose only, say at 23 degrees centigrade, and then we can like screen for temperature sensitive mutations by replicating them, one at 37 degrees and one at
You
23
are
degrees.
assuming that
So I was like thinking.
You are assuming
I'm
that
sorry,
the
sir?
mutations. You are assuming that the
Yes.
mutations will be temperature sensitive. It may not be the case,
Yes.
right?
Yes, that is also possible. But then, like, since there are so many mutations that are happening in the yeast cells, then we can expect some temperature-sensitive mutants in
Can I
galactose
try
metabolism,
answering
right?
this?
Yeah, Ankit, go ahead.
Yeah, so the thing is, like, for any experiment we do, like, we are seeing the galactose plate, but we have to compare the galactose plate with some things to know that if they are mutants or not. That is why, like, on glucose, we know that 100% every cell will actually grow. Okay, and then when we compare this glucose plate and galactose plate, then we can compare and see, okay, these are the mutants. We can actually identify the mutants. Without a mother plate, how you will identify the mutants?
Yes. So, to give an analogy, imagine that I want to identify those students who played Holi the day before. And I called all the students to the classroom, and the security ensured that no one with color on their body can enter the classroom. I will not be able to detect any one of you who played Holi the day before, right? So this is about gating, that if you plate it on galactose containing medium to begin with, then those that cannot grow in presence of galactose will not be able to grow. So you have no option of identifying those mutants.
Okay.
You have to plate them on glucose first, where, as Ankit rightly said, that every cell will have an opportunity to grow. Then you replica plate it on a galactose containing plate, and those which are defective in galactose metabolism will not be able to grow. Then you compare the original and the replica and see which are the colonies that are present in glucose containing plate but absent in the galactose containing plate. Those are, I like the language used by Shubhajit, likely or maybe mutated in galactose metabolism. Okay? Yeah, Ubaid, you were trying to say something.
No, sir. I was just adding some points that you add some more points, Ankit, in the follow.
Okay. Shubhajit, you had something to say.
Yes, sir.
Ah.
So in the suppression analysis, I have gone through the paper of art.
We will come to suppression analysis a little later, okay?
Okay.
You know, like as you can imagine, that having 1 hour discussion class per week is not going to serve the purpose. Okay, so I need the class to discuss among themselves and quickly give me at least 2 additional hours of discussion, which should preferably be in the evening, so that we have little liberty of having longish discussion hours. Because 1 घंटे में तो काम बनेगा नहीं. Like you will all, this is a very adult subject. Okay, this is not going to be. You will need me. You will need me to understand it properly. Okay, so Shubhajit, we will come to your suppression thing later. Piyusha, we will come to your question even later. But Piyusha, now let me ask you.
Yes, sir.
That you have compared the glucose containing plate and galactose containing plate, and you saw that some of the colonies could not grow in galactose, but they grew in presence of glucose.
Yes.
What will you do now?
Sir, now we know that this colony which is growing in the galactose and some are growing in the glucose, but maybe these ones which are growing in the glucose, if we take some from them, they will not again grow in the galactose.
Piyusha,
Yes, sir.
you claim that you have been trained in engineering subjects, right?
Yes, sir.
Please use precise language. What you are saying, some, if, they, that is absolutely a disaster. Use very sharp language. And Piyusha, I am telling you again, if other friends of yours are spending 9 hours per week for this course, you will need to spend 18 hours per week on this course. And it is not just about thinking, it is about reading. When you are having difficulty, immediately sending me a WhatsApp message. You can ask your classmates. Whoever has sent me a WhatsApp message, I have promptly responded to it. If necessary, I will have a special class just for you. But the standard will be the same. I will hold you at the same standard as anyone else in the class. Okay? So your language is not appropriate. I will give you a start with the language. That I will look at the original glucose containing plate and the galactose containing plate, and I will observe which are the colonies that have grown in presence of glucose but not in presence of galactose.
Yes.
Now start from here.
So we have a plate containing yeast growing in the glucose plate and another who are growing in the galactose plate. So now we know that the yeast who are mutated, they cannot grow in the galactose portion, but they can grow in the glucose portion.
Very good.
Yeah.
It is not
so,
portion, these are separate plates. Okay.
In plates, yes. So after that, we will, sir, go for temperature screening method. So in temperature screening.
Okay. Any statement you make, the answer will be why.
Okay. So, sir, first screening was to get an answer that the mutated portion, mutated plate, which were growing in the glucose, but not in galactose. Now there can be another mutated plate, portion of the colony, which are growing in the permissible temperature of 23 degree, but not higher than that in 36 degree. So the next screening can be done in the temperature sensitive screening manner.
But what will happen to those which did not grow in presence of galactose even in 23 degrees centigrade?
So sir, we knew that those are the portion which are not even growing in the 23 degree permissible temperature, as their enzyme, which is regulated by the gene, which is there for the usage of galactose, had been mutated and not working. So that portion we have screened already using the galactose and the glucose screening portion. So, sir, are you asking?
No, your answer is not correct. You try to answer it again, that there are certain. So do you know how yeast cells grow on a plate? Have you looked at in Google? Have you tried to do an image search of yeast cells growing?
Yes, sir, I have looked, and I have also looked for the spores, like how they are becoming spores in like.
How do yeast cells grow on a plate?
Sir, like colony and how to answer? I didn't got it like.
No, how do they look like? If you have to draw a plate where yeast cells are growing, how will you draw it?
So they are like dots over a plate.
Very good.
Like
so they're like
shown in
dots
the
over
slide.
a plate, right?
Yeah.
So certain dots are missing in the galactose containing plate.
Yes.
First of all, you answer one question in your mathematics language.
Yes, sir.
The dots that are present in the plate having only galactose.
Yes.
And the dots that are present in the plate having only glucose.
Yes.
What is their relationship? The dots that are present in the galactose containing plate, are they a subset of the dots present in the glucose containing plate, or will there be some that are mutually exclusive, or they will have no such relationship?
So I feel they will be like subsets.
Right? Subsets or subset?
Subset.
Subset. It is one-to-one subset relationship, okay?
Okay.
So now you see some dots are missing.
Yes.
Now, why should you need to go for a temperature, changing the temperature? They are already unable to grow in presence of galactose, right?
Yes.
Like by changing the temperature, what will you change? Like if you want to go for temperature sensitivity, Which plate will be your starting point? The glucose containing plates or the galactose containing plates?
The glucose containing plate.
Why?
Sir, we already know the ones which are not growing in the galactose medium. So first, we will
stop there. Why are those cells not being able to grow in galactose medium?
Sir, there is a mutation in the galactose producing gene. Galactose, not galactose producing, but the enzyme.
Galactose metabolism pathway.
Galactose metabolism pathway regulating enzyme is not producing due to the mutation in the specific gene.
Do not over-interpret a data ever in life. Okay, not for the course, not for actual life, actual lab. All you know is that these cells cannot grow in presence of galactose. So you say that they are defective in galactose metabolism. It is not necessary that they have a problem with an enzyme. Okay, there can be 300 other reasons why they are not able to grow. Okay, so fine. So they have a defect in galactose metabolism. Now what will you do? So now why do you need to again do temperature sensitivity assay?
So now I want to see if they are also mutated in a way where they cannot grow in the higher temperature, which was normal in wild type.
Okay, let's do some names. Okay, just hold on a minute. Okay, I am sharing my screen. Okay, so this is the original plate. Just to make my life simple, there are what, 1, 2, 3, 4, 5, 6, 7, 8. 8 colonies, okay?
Yes.
This is glucose. And Shubhadeep, right?
Shubhajit, sir.
Shubhajit. Shubhajit, actually, this is how it is done, okay? You keep the original plate intact.
Yes.
You make 2 copies out of that.
2 copies, yes.
Okay? You see, like 2 colonies are missing now, 6 and 7.
Yes.
Now you tell me, which plate would you go with? 1, 2, 3. Which plate would you go with for temperature sensitivity assay?
So for the galactose, that
I
is
have
2.
number 2.
Yes.
So if you go with number 2, what will you do? So should I now replica plate this, or should I do what? If you want, I can make a replica plate of this as well. Should I do that?
Yes, sir. So
Okay. I am doing replica plating for you. Whatever you say, I will do that. Now what do I do?
Sir, I think I have done a mistake here. So this galactose plating, before doing the galactose plating, we have preserved the colony in glucose media. So we already know which are the portion which mutants are not growing in galactose media, but which are growing in glucose media. So in the glucose plating, those mutants which have grown in glucose media, we should go for a temperature screening for those colonies first.
Like, just hold on. Meaning this one and this one?
Sir, one, yes.
Huh?
So the one is the replica plate before the galactose media. Okay, so yes, that one.
But these two colonies are not growing even under normal temperature. Why do you want to torture them on top of that with a high temperature?
Sir, this plating.
See, no. See, like, let us try to work on the fundamental question. What was the question that you started with? You started with the question that I want to identify genes that are responsible for yeast growing in presence of galactose as the only carbon source. Isn't that true?
Yes.
So, does these two colonies grow in presence of galactose? 6 and 7?
No.
So are they, therefore, is it not at all possible that they are mutated in genes responsible for galactose metabolism?
Yes.
Yes, meaning what?
So they have mutated such that they are not growing in the carbon source where the galactose is only present. Yes. So
does it not address the question that you started with?
Yes, it addressed the question.
Then why do you want to go do something else again?
Okay, I got shifted from the main question. Sorry.
Do not, memorize things. Always go. It is a 100% logical approach. Okay. If the logic fails anywhere, you alert me. There is no like two way of looking at it. This is the logic, and that's the only logic. That you wanted to set out to learn which are the genes that are necessary for yeast cells growing in presence of galactose. You randomly mutated, and you found that there are two colonies that are not growing in presence of galactose, but they are growing in presence of glucose.
Yes.
Just the fact that they are growing in presence of glucose means that every other apparatus is likely to be all right. It is only their ability to grow in presence of galactose as the sole carbon source is likely to be affected.
Okay, sir.
Now, what do we do?
Sir, so the temperature screening method will show us if they also grow in higher temperature or not. But the carbon source problem is solved, I guess.
Well, temperature screening, I will come to that later. First, you tell me that you have identified these two colonies that mutated in a manner that they cannot metabolize galactose. What will you do now? And this is only two, right? You have 1,000 such plates.
Yes.
Do you realize that this is only plate number 1?
Yes,
you have
sir.
1,000 such plates.
And so these are all in haploids.
All in haploids. And for every haploid, you have created these two replicas. So you started with 1,000 plates, you now have 3,000 plates. 1,000 original and 2,000 replica. Now what are you going to do? So this kind of colonies that cannot grow in presence of galactose, maybe 1,000 you have. Now what are you going to do? Okay, so we will end this discussion hour right here because you have another class. We will start right from here. I have only this request to make, that please come back quickly with 2 time slots, preferably in the evening, but I do not care. I am available 24/7, where we have 2 additional discussion hours per week. And you fix it and inform every other instructor so that we all know, like, this is the problem that is likely to be faced in every course. So you have only 4 courses, right? So I am confident that you will not be having more than 4 hours of direct contact with any of your professors. So 16 hours in a week. Okay, while in a week you have 24 multiplied by 7 hours. Okay, even if you live between 12 midnight to 6 a.m., you will still have 18 multiplied by 7 hours. So I'm pretty sure it can be scheduled in a manner that there is no overlap. Okay, so please come back to me by the end of the day today with 2 time slots that will be hard coded in our program, that you will inform all your other instructors, and I will put it on my diary so that we meet every week 3 times. Okay, otherwise you will not be able to appreciate this course or fully learn, fulfill the objectives of the course. Is that okay?
Yes, sir.
Yes, sir.
Okay. So please get back to me with 2 slots by the end of the day. Okay. And I will share the recording of this soon. I find it much easier to put things in YouTube, and the instruction is also that it is better to do it on YouTube because those who do not have very good bandwidth, they might find it easier to hear it from YouTube than on Hello IITK. But I will put it in both the places. Okay. Okay, bye. I'll wait to hear from you. Bye-bye.